Welcome viewers. Today I am going to explain about the inner ear, that is the cochlea, along with the organ of corti, the cochlear nerve and the other important related structures. The cochlear nerve, also known as the acoustic or auditory nerve, is the cranial nerve responsible for hearing. It passes out from the inner ear through a bone located on the side of the skull called the temporal bone and finally enters the brainstem at the level of pontomedullary junction. The cochlear nerve is purely a sensory nerve. It has no motor or movement function and is one of the two parts of the vestibulocochlear nerve, also known as the cranial nerve 8, and the cochlear nerve is responsible for hearing. The vestibular nerve is responsible for balance, motion and position. The inner ear lies within the bony temporal bone and can be divided into two parts, the bony labyrinth and the membranous labyrinth. The bony inner ear can be divided into three parts, that is, the semicircular canal, the vestibule and the cochlea. The cochlea is made up of three canals wrapped around a bony axis called the modulus. These canals are the scala tympani, the scala vestibuli and the scala media or the cochlear duct. The scalae tympani and vestibule are filled with perilymph and are linked by a small opening at the apex of the cochlea called the helicotrema. Between the scala media and the scala tympani is a structure called the organ of corti. Now, let us see the orientation of cochlear nerve into the cochlea. The fibers of the cochlear nerve originate from an aggregation of nerve cell bodies, the spiral ganglion located in the modulus of the cochlea. The neurons of the spiral ganglion are called bipolar cells because they have two sets of processes or fibers that extend from opposite ends of the cell body. The longer central processes of the bipolar cochlear neurons unite inside the spiral ganglia and are twisted like the cords of a rope to form the cochlear nerve trunk, also called the primary auditory fibers. These primary auditory fibers enter the part of the brainstem called the medulla oblongata. The shorter peripheral fibers extend to the bases of the inner and outer hair cells, a series of tiny holes beneath the inner hair cells. At this point, they lose their myelin sheaths and enter the organ of corti as thin unmyelinated fibers. There are about 30,000 of these fibers and the greater number of them, about 95%, innervate the inner hair cells. The remainder cross the tunnel of corti. The tunnel of corti is the fluid-filled space, approximately triangular cross-section, formed by the inner and outer rods of the corti and the basilar membrane, extending the length of the organ of corti from base to apex. Here, you can see the three canals of the cochlea, the scala tympani, the scala media or the cochlear duct and the scala vestibuli. I would like to discuss the scala media in detail as it is the most important canal of the three canals. Filled with endolymph, the cochlear duct ends blindly at both ends, that is, below the round window and at the apex. In cross-section, this duct resembles a right triangle. Its base is formed by the osseous spiral lamina and the basilar membrane. The side of the triangle is formed by two tissues that line the bony wall of the cochlea, the stria vascularis, which lines the outer wall of the cochlear duct, and the fibrous spiral ligament, which lies between the stria and the bony wall of the cochlea. Now, let us see some of the structures inside the floor of the scala media. On the inner side of the inner hair cells and the cells that support them is a curved furrow called the inner spiral sulcus. This is lined with more or less indifferentiated cuboidal cells. This is the inner hair cells, which are the main type of receptive cells found in the cochlea, and they are responsible for converting sound waves into nerve impulses. The sound vibrations come through the fluid of cochlea. At the apical surface of the inner hair cells, both the stereocilia and one longer stereocilia called the kinocilium are present. There are approximately 100 teeny stereocilia. The kinocilium is present at one side of the inner hair cell. Both stereocilia and kinocilium face the tectorial membranes of the cochlea. The adjacent stereocilia are connected to each other by protein filaments at the apex of the stereocilia. Outer hair cells are other type of hair cells present in the cochlea. The sensitivity of outer hair cells are around 50 decibels. Therefore, the main function of the outer hair cells is to pre-amplify the sound waves with a low amplitude. Each outer hair cell is supported by a phalangeal cell of dieters or supporting cell, which holds the base of the hair cell in a cup-shaped depression. Now you see the outer spiral sulcus. The floor of the outer sulcus is lined by cells of epithelial origin. This region appears to have an absorptive rather than a secretory function, and it may be involved in removing waste materials from the endolymph. The secretory structures of the cochlea, including stria vascularis, spiral ligament, and supporting cells. Now you see the tectorial membrane. The longest stereocilia of the hair cells make contact with but do not penetrate the tectorial membrane. The hair cells in the stimulated regions are excited by the resulting shearing action between the stereocilia and the tectorial membrane. This membrane is an acellular gelatinous structure that covers the top of the spiral limbus as a thin fibril layer, then becomes thicker as it extends outward over the inner sulcus. TM is a highly porous, hydrated, and dynamic structure. The proelastic, electrokinetic, and wave properties of the TM are frequency and place dependent and thereby may be essential for cochlear sensitivity and frequency selectivity. Now, here you see the osseous spiral lamina, which extends externally in a concavity, the inner spiral sulcus, which represents on section the form of the letter C, the upper part formed by overhanging extremity of the spiral limbus as labeled, is named the vestibular lip, and the lower part prolonged and tapering is called the tympanic lip, and is perforated by numerous foramina for the passage of cochlear nerves. The scala media, filled with endolymph, is surrounded by the vestibular or Reissner's membrane on the side of the scala vestibuli and by the lateral wall composed of the stria vascularis and the cochlear promontory. It is separated from the scala tympani by the basilar membrane. The organ of corti rests on the basilar membrane.